So today we are performing Plasmid prep from QIAprep Miniprep Kit, and this is overnight grown culture which I have done the pelleting at 6000 rpm for 10 minutes at room temperature. Now I'm going to add solution 1 into it. So this is a 5 ml pellet of 5 ml culture. So according to the protocol, I'm going to add 250 microliter of solution 1, which has RNase A. So this should be kept at 4 degree. After adding solution 1, we have to mix the pellet in solution 1, and then we need to transfer this pellet in a mini centrifuge tube. That I'm going to add, this is solution buffer P2, this is lysis buffer, and I'm going to add equal volume of solution 2, which is 250 microliter. After adding solution 2, vigorous mixing like pipetting or vortex not allowed, so we have to do mixing by inverting the tubes several times, so it will lyse the cells. And after adding solution 2 and mixing, the solution will become more clearer than before. We can see that this is little bit clearer than before. Now I'm going to add, this is buffer N3 neutralization buffer, and this is according to protocol. For 5 ml culture I have to add 350 microliter of solution 3 in that mixture. Again, after adding solution 3, I have to mix it by several times inverting. It will form a cloudy white cloud formation. After mixing it, we have to pellet down the cell decrease at 14,000 rpm for 5 minutes at room temperature. I am setting the centrifuge at 14,000 rpm for 5 minutes at room temperature. जैसे स्टार्ट करो नहीं बन रहा है. So after centrifuge, whole debris got pellet down. This is Qiagen Miniprep column. from which we have to pass the supernatant which contains plasmid. So in one go we have to add 750 microliter of supernatant, and then after that we have to pass it through the column. So for that we need to apply some centrifugal force, and for that we need to centrifuge it for 1 minute at 40,000 rpm again. So it's done. Whole supernatant get passed through the column and collected in the bottom tube. I discarded the collected solution, and now I'm going to add the wash buffer, which is PE wash buffer, and it contains ethanol. I'm going to add 750 microliter of PE buffer to wash the column. Further centrifuge it at 14,000 rpm, so 1 minute to pass it through the column. Discarded the collected tea buffer, and now I'm going to centrifuge it, doing empty centrifuge for 5 minutes at room temperature, so that all the remaining ethanol will be passed through the column. After that we will keep that tube over a new append of tube. Kept that tube over a new mini centrifuge tube, and we will wait for several minutes so that remaining ethanol will evaporate from the tube. After 10 minutes of incubation at room temperature, so that all the remaining ethanol will evaporate from the tubes, I'm going to add 50 microliter water in the tube, in the column, so that to elute the DNA. And this is pre-warm water I have kept in dry bath, and I am adding 50 microliter water at the center of the column. I'll wait for again 5 minutes and then spin it down. After 5 minutes I'm doing the centrifuge for 1 minute at 14,000 rpm. So after that I will repeat the previous step again for one more time. I'm adding 50 microliter water again to the bottom of the center of column. I will keep it for 5 minutes. So I'm again doing final centrifuge. After that I will check the concentration of isolated plasmid by nanodrop. I'm going to check the concentration of isolated plasmid by nanodrop. Before that I have to set the blank with water. I have to nanodrop nozzle, and here I have added 1 microliter of water, which will be, this is the blank button. Checking the concentration of DNA. So this is the concentration of isolated plasma, which is 97.4 nanogram per microliter, and this is 260 by 280 ratio, and this is 260 by 230 ratio.